All right, everybody, welcome to Traits in Reproduction Lesson 2.2, Gathering Evidence About Genes. Now, for this activity, or this lesson today, there are some activities that would be a lot easier if we were together. But since we're not, I'm going to, again, ask you to get creative. There was a point where I asked you to come up with your own types of models that represented the proteins that connected in order to form spider silk. And I had some really cool stuff that people sent me, some pictures that people sent me of Legos and like pencils and rubber bands and paper clips, things like that. Cool creative stuff. And I will be asking for the same thing again later on in this lesson for you to come up with examples and models. And you can send them to me. But first, before we do any of that, let's go ahead and get to the warm-up, which says, let me go ahead and just pop this open in Amplify. It's asking you about a protein that we saw. Ooh, review the diagram below from the Hemophilia Proteins and Genes article. Remember, we read that last lesson. Oh yeah, I remember that. There are 13 different clotting factor proteins. This model represents a molecule of one of those proteins, the one most hemophilia patients are missing. So this is supposed to represent a protein. Pardon me. Oh man. Oh, how many of you just yawned? Huh? Why is that? I don't know. That's not what we're learning about today. Genes provide instructions for making clotting factor proteins. The diagram to the left is an example of just one of hundreds of thousands of different proteins made in the cells of your body. What other types of proteins do you think your body makes? List your ideas below. Great question. What types of proteins do you think your body makes? Go ahead and answer that question and come back to me. All right, now let's talk about this activity that we were supposed to have done together, which some of you still could totally do at home if you find some materials to do it. So here's our investigation question for today. How can organisms make different protein molecules for a particular feature? How do they actually make these different protein molecules? Well, in the article that we read yesterday, what other types of proteins do you think your body makes? This is related to our warm-up question. We know that we make these clotting proteins, and maybe even think about like, okay, well, in the simulation, we saw that spiders made these types of proteins. What about you? What about you? Here's one of our key concepts. Genes are instructions for proteins. This is something that we learned from reading the article yesterday, that the genes that we have in every single one of our cells code for specific proteins. They're blueprints for proteins. So our cell knows exactly which proteins it is supposed to make. So our investigation question asks how organisms can make different protein molecules for a particular feature. We're going to model this in just a second. We know that organisms can make different protein molecules because they have different genes. Now we need to figure out, well, what role do genes actually play in the making of proteins? Because we actually had talked about some claims previously with genes. What we were going to do is we were going to, as you can see in this lovely picture, we were going to stick our tongue out and try to put these connect pieces together. Sort of. Actually, I'm sure that's how a lot of you would look in class if we did this. But today we will use physical models to learn more about how proteins are made in the cells. Let's look at two claims about the role that genes play in building proteins. We're going to decide which claim we think is more accurate by the end of this. The first one, genes provide instructions and build proteins. Okay, hey, let's look at these things over here. Genes are located in the nucleus of the cell. Everyone say, by the way, nucleus, nucleus, nucleus. Thank you. This is where we find our genes. Okay, we just did a tiny bit of study in microbiomes on certain features of the cell, and hopefully we remember that the nucleus was something in the cell. The nucleus is where we find all the genes. That's where all of the blueprints are located. Genes contain instructions for building specific proteins. Fantastic. So they contain these instructions, and genes, here's the claim, genes use the instructions to build specific proteins. Genes provide instructions and build proteins. Well, here's our other claim. Genes provide the instructions for making proteins, but they do not build the proteins themselves. So the first one's saying that genes have the instructions and genes actually put them together. The second one's saying, no, genes just have the instructions, but someone else puts the proteins together. So here's what we would have been receiving today. You would have received all of those pieces that you see in front of you. And in your nucleus, it would have said A1A1, A1A2, or A2A2. And you would have had instructions inside of the bag. The pieces, all of the little individual pieces, represent the building blocks of proteins. What are those called? Oh yeah, amino acids, right? That's what we said. The envelope represents the nucleus, which contains genes. So the nucleus has all of the information, all of the genes inside of it. And there are instructions inside each one of the envelopes. And what you would do is two people, if we were in groups, would read the instructions. There's two instructions in each of the envelopes. Two of you would read it, and then the other two people would try to follow the instructions and build their genes, okay? Ultimately, here's what it would look like. Not that, but this. Some of you would have had instructions for two A1 molecules, and you would have had two of these over here on the left. Some of you would have had instructions for two A2 molecules. Some of you would have had instructions for one of each. Just luck of the draw. But that's ultimately what we would have ended up building. So in Amplify, there are some questions actually for you here. Hang on one second. Let me get back to this. I found the droids I was looking for. Actually, it's just in lesson 2 right here, or activity 2. In the modeling activity, what do each of the items represent? We actually just talked about this, didn't we? What did the envelope represent? Oh yeah, it represented the nucleus. The instructions, those were the genes, and the students building the protein molecules. This one may have escaped you, may have passed you by. The ribosomes. The students building the protein molecules were the ribosomes. That's actually where the proteins get put together in the cells. So compare your two protein molecules. Are they the same? Are they different? Or why? Remember, you didn't actually get instructions, but you can make it up. The most important part is why. If they were the same, why were they the same? Because each person had the same instructions. If they were different, why were they different? Each person had different instructions for the proteins. So which claim about genes' role in building proteins best fits considering the modeling activity you've just completed? Did the genes themselves actually build the proteins? And we should have realized, no, the genes don't. They have the instructions, but they don't actually do the building. The ribosomes do the building. That's where the building takes place. So claim number 1, we could say, no, not really. It's claim number 2. Genes provide instructions for making proteins, but they do not build the proteins themselves. Let's go back to the slides. We'll jump right here. Some groups built 2 copies of the same protein, while some groups built 2 different proteins. Why might this be the case? We already talked about this, that they had different instructions, right? So this leads us, though, to our key concept number 6. Each gene version provides a unique instruction to make a specific protein molecule in an organism's cell. So we have these gene versions, and they have specific instructions, and each instruction makes a specific protein. Okay, I gotcha. Here we go. Gene version. Good vocabulary term here. A specific form of a gene that provides instructions for making a particular protein molecule. We also might end up calling this an allele, which we actually probably will right here. Genes are instructions for making proteins, which determine the traits of an organism. There are different versions of each gene. Each gene version is a unique instruction for making a specific protein molecule. Different versions of a gene can also be called alleles. So let's kind of zoom back out here really quick. Let's think of our spiders. Our spiders had genes for silk flexibility, right? Now, we saw that the spiders, in that silk flexibility category, had sometimes different proteins. Their cells would make different proteins. Sometimes they make the same proteins. Each of those proteins has a specific set of instructions, and each of those is called an allele or a gene version. Okay, so the different gene versions can also be called alleles. All right. Now let's talk about mutations. There is another modeling activity for this with our physical models, but we will try to navigate around it. First, what do you already know about mutations? I'm going to ask you this question when I see you. Hey, what do you know about mutations? I mean, for me, green glowing mutagen that turtles crawl into and they become Leonardo, Michelangelo, Donatello, and Raphael and Splinter. That's what I would have known about mutations when I was in elementary school. Of course, I know a lot more now. You will now model what happens when there is a mutation in a gene. You'll use the same model as before, but this time you'll add in 2 new steps. So what actually happened before was you would have received 4 steps, like how to make your model. They'd look like this, just like this. So you would have received these instructions: step 1, step 2, step 3, step 4. If you were making gene version A1, if you're making gene version A2, you would have received these instructions: step 1, step 2, step 3, step 4. Here's how we are showing mutations. You would have received another strip of paper, okay? And it would have had step something on it. Here are the mutations. You may have had any one of these, mutation A, B, C, D, or E. You would have only gotten 1 strip of paper. And whichever step it says on it, step 1, 3, 4, 3, you would have taken that strip of paper and placed it over that step. So you no longer would have done the original step. You would have done one of these for whichever step is labeled step 1, 3, or 4. That can cause, as you would have seen, all sorts of problems. Sometimes you get to the next step and it just doesn't make sense, and there's nothing that you can do. Where you might have, I don't know, 2 rods like this, and it says, now connect them. Like, well, wait a minute, they don't. And all of a sudden, your protein just doesn't work, and you cannot even make the protein anymore. Other times, you are able to make a complete protein, but it's different. It is not the same structure as before. If it doesn't have the same structure, is it going to function in the same way? That's a good question. So the result of adding a mutation to the gene was that we should have noticed we were not able to make the same protein. The protein itself was different. And hey, that makes sense, right? If you change the instructions, you get a different final outcome, okay? Vocabulary term: mutation, a random change to a gene that sometimes results in a new trait. I want to highlight the word random because mutations are always random. Now, we've seen lots of movies, right, where people mutate their genes, or they mutate because of a need that they have, right? Like, oh, this person developed gills because they live in a water world, and that makes sense because they would need gills. Well, wait a minute, Kevin Costner. That's not how mutations work. Mutations are random, completely random. If someone ended up with gills, hey, great. It's not because you live in a water world. It's just because something got messed up in your genes. You may live in a desert and get gills, and it doesn't even work like that. I'm just saying. It's not based on a need that an organism has. It's random. Anyways, not to get into all of that, but it is random. Okay, that's all I'll say. That sometimes results in a new trait. It doesn't always result in a new trait. Sometimes it's just not even functioning. In a mutation, the gene version changes. That means the instructions to build the proteins change, which can cause the protein to be shaped differently. Mutations that affect the trait of an organism are actually very rare. Most mutations result in a new protein that does not affect a trait because it does not interact with other proteins. Very rarely does a mutation occur with instructions for a protein that will interact and affect a trait. It is extremely rare. And it's even more rare for that protein or that new trait to be even beneficial for an organism. So there's some questions for you to answer in activity 3. I'm getting to them right now. Record your observation. That's it right there. The change in the instruction caused the protein to, hey, I'll just go ahead and give it to you. It's the top 3. But maybe yours was different because it would be specific to what you had. And let's go on to the homework. The homework. You get to watch a cat video. Lucky you. So you're going to watch a video about cats. It's attached to this lesson. It's also on my YouTube channel. And then I want you to answer this question. How can a mutation result in a new trait? Watch the video. It's cute. It's cute. You'll love it. Anyway, that's it. I'll see you later.